Hello friends. So after completing the topic of spermatogenesis, that is the formation of functional haploid sperms, we will start with the topic of structure of sperms. So let us begin with the topic. Now in the structure of sperms, you can see that it is basically divided into three main parts. The sperm has three main parts, that is head, middle piece and tail. Now this is the head portion, you can see over here. Now the tip of the sperm, you can see over here, this acrosome is the tip of sperm. And what is this tip of sperm consisting of? This tip of sperm is consisting of enzymes that generally breaks the wall of ova. Okay, so that enzymes are hydrolytic enzymes, generally proteolytic in nature. Hydrolytic enzyme called, or you can say digesting enzyme, okay, or digestive enzymes called as hyaluronidase. Hyaluronidase. Okay, so it consists of enzyme called hyaluronidase that will digest the wall of ova during the fertilization. So that is all about the first point in the head portion, that is what you call it as acrosome. Alright, and then comes the nucleus, right? Over here, the nucleus consists of condensed or concentrated DNA of father or DNA of the male, you can say like that. So it is the area which is rich, which is concentrated, which has a lot of like whatever the amount of DNA a father has in a haploid genome form, that is present over here in the nucleus. So this portion is what you call it as the. So this portion is forming the head. So what does head consist of? Head consists of mainly two structures, that is the tip called as acrosome, that consists of digestive enzyme, and that digestive enzyme is here called as hyaluronidase, that digests the wall, the outer layer of the ova during fertilization. Now next is nucleus. We have seen that it consists of condensed form of, you can say, the male DNA or the father's genome. Then comes the next, that is known as the middle piece. Okay, now this middle piece is comprising, you can see a wavy structure over here. So this middle piece is consisting of, it consists of mitochondria. Okay, it consists of mitochondria. Because we all know that the male gamete is motile, so it needs a lot of energy for the same motility. So who will provide that motility, or who will provide the energy for that motility of that sperm? Because when it is going to get ejaculated in a female body, it is going to travel till fallopian tube. So who is going to provide that motility, that active motility or energy for that motility? That is being provided by this mitochondria, which is located in the middle piece. So over here, it provides energy in the form of ATP for motility. Remember this. Okay. Now next is this tail. You can see this. It is a tail. Now this tail is for what? It is giving that lashing movement. So because of its tail, you can say that the sperm is able to move. So this sperm, and it is composed of centriole. Okay, it is composed of centriole, and it provides motility. Or you can say that it is the one which helps in the movement. You can say that it acts like a flagella, the way unicellulars have got flagella, which is responsible for the motility of that cell. Here it's the tail which is responsible for the motility of the sperm. So ideally we can say that the sperm is divided into three main regions: the head, the middle piece, and the last one is the tail. Okay. Now if you talk in context with the fertility of the male, let me tell you that a male ejaculates 200 to 300 million sperms per ejaculation. That means what? That during one single intercourse or coitus, a male can release 200 to 300 million sperms. Isn't that a massive number? Yes, of course, right? And then onwards, we can say that we have seen the motility. So what determines the motility? Let me tell you that all of these sperms, that is the number is 200 to 300 millions per ejaculation. From that, 60% of the sperm should be must be actively motile. It should have that speeded motility. Then and only then we can say that that particular male is fertile or can reach the ova and can fertilize that ova. So this is something a really important point. 200 to 300 million sperms per ejaculation, from which there should be 60% of motility, 60% of the sperm should be motile, and that demarks the we can say that the fertility or the fertilizing factor, or whether the male is fertile or not, that has been determined by this motility. So this is all about the structure of sperm. Now we'll start with the topic of hormonal influence on the spermatogenesis process. We all know that the process of formation of sperm starts occurring in male at puberty. So exactly what happens in the male at puberty that this process starts. So that is all about the next topic, that is hormonal control over the spermatogenesis process. So here we go with the process. We know that there is one structure, in that is what we already studied in a chapter called chemical control and coordination. That hypothalamus has an effect on pituitary gland, and then pituitary gland being the master gland of the body, it is influencing the other glands of our body. Right? So over here also the same process has to be considered. Now here, how does the spermatogenesis start? So hypothalamus starts releasing the hormone called gonadotropin releasing hormone. Gonadotropin, GnRH. So it is going to have an influence, so it acts on the anterior pituitary gland. We also call it as adenohypophysis, right? So it acts on anterior pituitary gland, also called as adenohypophysis, right? And anterior pituitary gland starts releasing a hormone called, two hormones it will release. The first hormone is called LH and the second hormone is called FSH. So here two hormones are released from the pituitary gland. The first is LH, which is also called as luteinizing hormone, and the second one is known as the follicle stimulating hormone. So as we are here, we are talking about spermatogenesis. So LH and FSH both will be having an influence on gonads, right? And in male, the gonad is testes. We all know that, right? So luteinizing hormone will act upon the Leydig cells of the testes. We know that the Leydig cells are also called as interstitial cells. They are also known as interstitial cells. And the function of Leydig cell is what? To secrete the hormone called testosterone, right? So over here, the first influence of luteinizing hormone is released from the anterior pituitary gland is what? That these luteinizing hormone will act upon the Leydig cell of the testes, that is also called as interstitial cells. These interstitial cells or Leydig cells will start secreting a hormone called testosterone. And this testosterone will influence or will start the process of spermatogenesis. Right? So this is how it is going on. This is how we can say that the formation of sperm starts in a male at puberty. Now further, if we talk about the function of follicle stimulating hormone. Now it again is released from anterior pituitary gland, that is the adenohypophysis. And it goes and acts upon the seminiferous tubules of testes. Once it goes and acts on seminiferous tubules of testes, particularly it has an influence over Sertoli cells. And these Sertoli cells will stimulate the process of spermiogenesis, particularly spermiogenesis. Now what is spermiogenesis? So we have seen in the previous class that spermiogenesis is a morphological differentiation of spermatids, that is a round cell, to the head tail wala sperm, that is this, right? That is what you call it as spermiogenesis. So over here it is doing the function is going on this way. So first point, we know that spermatogenesis starts at puberty. The gonadotropin releasing hormone is released from the hypothalamus. Hypothalamus releases that gonadotropin hormone, which goes and influences the anterior pituitary gland, which further influences which releases the LH and FSH. Now what is LH? LH is luteinizing hormone and FSH is follicle stimulating hormone with the respective function of spermatogenesis and thus spermiogenesis. So this is all about the topic of hormonal influence over the spermatogenesis process. Next topic to understand is the sectional view of seminiferous tubule. Let us see how does the sperms are produced. So that is what we are going to talk here about. Now, so we talk about the sectional view of seminiferous tubules. In the previous topic, we have already studied about the topic of spermatogenesis, in which we have seen the different phases of sperms. So now let us begin with the topic of sectional view of seminiferous tubules. Now let's say that this is the germinal epithelium of seminiferous tubules. So I'll mention this as a germinal epithelium. Now what is this germinal epithelium composed of? This germinal epithelium is composed of cuboidal epithelium, right? So let's say there are cells over here which are cuboidal epithelium. Now these keeps on dividing and they keep on germinating, so we call it as germinal epithelium, and they produces a cell, we call it as spermatogonium. So they divide by mitosis and produces many cells with a profound nucleus inside it, and these cells are called as, the one which comes out from the germinal epithelium are called as spermatogonium. Remember that spermatogonium is diploid cell, right? So here it will be having 46 numbers of chromosome. Spermatogonium keeps on dividing by the process of mitosis again, and it produces primary spermatocytes. So after this, mitosis again occurs and it starts producing cells called primary spermatocyte. So this is a sequence of cell, we call it as primary spermatocytes. And this is also being produced by which division? The division called as mitosis. So this is also going to be 2n only, and 2n is what? 46 numbers of chromosomes are present over here. Then it grows and it becomes large, but still it is primary spermatocyte only. Further, it divides by meiosis. It divides by meiosis and produces other cells called secondary spermatocytes. Secondary spermatocyte. Now as we know that meiosis has taken place from primary to secondary. So from 2n it is going to get reduced, and that's how it will be forming n, right? So here this secondary spermatocyte will have n number of chromosome, and that is what? 23 numbers of chromosomes. So let us draw some nucleus inside over here. This will again divide and produces spermatids. So this is spermatids, and it divides by meiosis 2. So spermatid is again, from n it remains n. N is equal to how much? 23 numbers of chromosomes. And from this, after the process of spermiogenesis, that is the morphological changes taking place, you can say that what is formed. So the structure that is formed is, we call it as the sperm. So this is what you can call it as sperm or spermatozoa. Because you can see that it has differentiated into head, middle piece and tail. So this is known as spermatozoa. Now, during the process of spermiogenesis, what is happening that once the sperm is formed, it takes nourishment from a particular cell that is present along with this cell, and that cell, if it is taking nourishment from that cell, I am drawing it with a different color so that you can predict it. So put this cell somewhere in between. Right? So this cell is known as the Sertoli cells. Right? And we already know what is the function of Sertoli cells? To provide nourishment to the developing sperms. So this Sertoli cells will provide nourishment to the developing sperms. Or this spermatids, you can say that this spermatids will acquire nourishment from the Sertoli cells and it will develop into the sperms. So this is all about the sectional view of seminiferous tubules.